Imagine you're sitting in an oncology waiting room, and there are two patients sitting right across from you. Let's call them patient A and patient B.
Right, just two regular people.
Exactly. And they both just received the exact same diagnosis. It's diffuse large B-cell lymphoma, or DLBCL.
Which is, a highly aggressive blood cancer.
It is. And because their medical charts look basically identical on paper, their doctors prescribed them both the exact same gold standard treatment.
Yeah, a really heavy-hitting chemoimmunotherapy regimen is called RCHOP.
Right, RCHOP.
It's the frontline defense. I mean, it's the standard of care across the board for this.
Okay, so they both go through the treatment. But here is where the mystery really begins. Patient A goes into full remission, like the cancer is completely wiped out.
Oh.
But patient B, on the other hand, relapses almost immediately. The tumor it doesn't seem to care about the RCHOP at all.
Yeah, and for decades, oncologists have basically been tearing their hair out trying to figure out why this happens.
Right. I mean, we've sequenced the DNA, we've looked at the genetics, but those genetic classifications just keep failing to tell the whole story.
Because we've been staring at an incomplete picture, We've been treating the starting line as if it's the actual finish line.
And that really brings us to the core mission of our deep dive today. Welcome, by the way. Today, we are thoroughly unpacking a truly massive paradigm shift in how we understand cancer.
Yeah, it's based on a groundbreaking 2026 research paper.
Exactly, from the journal Cancer Cell, published by Eslil and colleagues. They basically decided to stop just looking at the genetics, and instead they used this incredibly complex new approach called multi-omics.
Multi-omics, yeah, to uncover the hidden functional identities of these tumors.
And what they found is a deadly, invisible variant that they've named PG4. We're going to break down every single concept, mechanism, and term today until all the details are crystal clear.
Sounds good. To understand how they actually unmask PG4, we need to quickly look at what we used to do.
Right, set the stage for us.
So historically, DLBCL was classified using a system called cell of origin, or COO for short.
That's COO, got it.
This was based on looking at the RNA of the cancer cells to try and figure out what stage of normal B-cell development the tumor originally started from.
So it's basically tracing the cancer's family tree. Like, did it originate from this specific type of cell or that type of cell?
Exactly. And that gave us two main buckets, ABC, which stands for activated B-cell-like, and GCB, germinal center B-cell-like.
Okay, ABC and GCB.
Yeah, and for a long time, we knew ABC tumors generally had worse outcomes. Over the years, we layered on genomic sequencing to find specific DNA mutations, creating these genetic subtypes.
But even with all that RNA and DNA data, we still had patients, like your patient B from earlier, who just didn't respond at all.
Right, because knowing where a tumor came from doesn't always tell you how it operates today.
Which is wild. So the researchers in this paper realized they were missing a massive piece of the puzzle, and they used multi-omic profiling.
Yeah, multi-omic.
We know genomics looks at the DNA. And transcriptomics looks at the RNA, but they added a third layer, right? Proteomics.
Yes, looking at the actual physical proteins inside the cell. And adding that protein layer is what completely broke the case wide open.
Let's build a mental model for this so we can really picture it. Think of it like a massive construction site.
Oh, I like that.
Yeah. So genomics, the DNA, is the architect's master plan. It contains every potential design for the building.
Right, and transcriptomics, the RNA, represents the paper blueprints that actually get printed and sent down to the foreman on the site.
Exactly. But proteomics, looking at the proteins, that is the actual bricks and mortar. It's the steel beams being welded together on the ground.
That's a great way to look at it, because sometimes the paper blueprint, the RNA, says, we need 100 bricks for this wall.
Right.
But down on the factory floor, a machine breaks, or a manager goes rogue, and the factory only produces 10 bricks. Or maybe it goes crazy and produces 1,000.
Right. And if you are an oncologist and you only read the paper blueprint, you are completely blind to the reality of the building. You literally have to step onto the site and count the actual bricks.
But counting the bricks at a microscopic level across thousands of cells, I mean, that is an absolute nightmare.
How do you even do that? I mean, physically?
Well, it requires some of the most advanced technology we have. The researchers took biopsies from 332 patients. That was their discovery cohort.
Okay, 332 people.
Yeah, and to count the proteins, they used a technique called TMT-based mass spectrometry.
Okay, stop right there. Break that down for me. What does TMT actually do to a tissue sample?
So TMT stands for tandem mass tags. Imagine you extract all the proteins from a tumor biopsy. and you chop them up into these tiny little pieces.
Chopped up proteins, got it.
You then attach a microscopic chemical barcode, the PMT, to every single piece. And this barcode has a very specific known weight.
Wait, so literally a physical weight?
Yes, a physical mass. So you can mix samples from dozens of patients together, put them into a mass spectrometer, and use lasers and electric fields to vaporize them.
Vaporize the protein.
Yeah. Vaporize and sort them by their mass. It allows you to precisely identify and quantify almost 7,000 different proteins per tumor.
Wow.
And you can do this simultaneously across hundreds of patients.
That is staggering. Okay, so now you have thousands of DNA mutations, thousands of RNA transcripts, and thousands of protein levels for 332 people.
Yep.
That is an absolute tsunami of data. Like, no human brain can just spreadsheet that out and find a pattern.
No, it's completely impossible.
Yeah.
Which is exactly why they used a highly advanced AI algorithm called MovieI.
MovieI, what does that stand for?
It stands for Multi-View Latent Variable Modeling.
Okay, latent variable, meaning like a variable that is hidden that we can't directly observe with our own eyes.
Exactly. Think of how a streaming service recommends a movie to you. It doesn't just sort by simple, basic categories like action or comedy.
Right, it gives you those super weird specific ones.
Yeah. The algorithm finds hidden, latent factors, like movies featuring gritty anti-heroes with dark synth soundtracks.
I watch a lot of those,
I'd
actually.
say the AI knows. And Movie does that exact same thing for biology. It overlays the DNA master plan, the RNA blueprints, and the protein bricks, and looks for hidden biological states.
So it essentially asks the computer, regardless of the starting blueprints, how many unique types of buildings are actually being built here on the ground.
Exactly.
Okay, let's take a breath here. We are about 5 minutes in, so let's do a quick recap of what we just covered before we move on.
Good idea.
Bullet point 1. DLBCL classification used to rely almost entirely on RNA blueprints, sorting tumors into origin buckets like ABC and GCB.
Yep.
Bullet point 2. Multi-omics is this massive shift where we look at the blueprint, but we also use TMT mass spectrometry and AI to count the actual protein-building material.
Spot on.
So listener, here's a question for you to chew on. Have you ever followed a recipe perfectly, but the dish came out totally wrong?
Oh, all the time.
Right. So how does understanding that gap between instructions and reality change your view of DNA? It's not just about what the DNA says, it's about what the cell actually builds.
Exactly. And when the MovieI algorithm overlaid those blueprints with the bricks, something shocking happens.
What happened?
Those old boundaries, the ABC and GCB categories we relied on for 20 years, they essentially vanished in the AI's eyes.
Vanished, like they didn't matter at all.
Well, the algorithm found that the 332 tumors naturally clustered into 7 totally new, distinct groups. They call them proteogenotypes, or PGs.
PGs, so PG1 through PG7.
Right. And these PGs completely ignore the old origin categories. They are grouped by something called convergent biology.
Convergent biology. Let's break that down. Does that mean tumors that started differently end up acting the same?
Yes. It's the realization that tumors might have completely different genetic starting lines. One tumor started as an ABC subtype, another started as a GCB subtype. But as they mutated, as they fought against the patient's immune system, they converged. They end up using the exact same biological tactics to survive.
Okay, let me try an analogy. It's like 2 criminals. One was a bank robber, the other was an identity thief. Completely different backgrounds.
Sure.
But eventually, they both realized the absolute best way to get rich is to run the exact same cryptocurrency scam. They converge on the same machinery.
That's a perfect way to look at it. They converged. And the most dangerous, deadly scam the AI found was proteogenotype 4. PG4.
Ah, the invisible killer we mentioned earlier. PG4, let's look at the sheer impact of this variant. How common is it?
PG4 makes up about 20% of all the DLBCL cases in this study.
1 in 5. That's huge.
It is. And the clinical outcomes for these patients on that standard RCHOP therapy we talked about, they were absolutely abysmal.
Really? How bad are we talking?
They looked at overall survival and progression-free survival. PG4 patients had significantly inferior outcomes compared to almost all the other groups. In fact, if a patient's treatment completely failed within the first 12 months, there was a 40% chance that their tumor was PG4.
Wait, I have to push back here for a second.
Sure ahead.
If PG4 just scoops up some ABC tumors and some GCB tumors and puts them in a new bucket, does that mean our previous decades of oncology and diagnostic categories were just a wild guess? Like, were they totally useless?
It's a natural question, but no, they weren't useless at all. They were just incomplete.
Okay, explain that.
Think back to your building analogy. The origin still matters. ABC and GCB tell us where the cell came from, like what neighborhood it was born in.
Right.
But PG4 shows us that an ABC tumor and a GCB tumor can independently evolve to build the exact same heavily armored, drug-resistant fortress.
Oh, I see.
When it comes to surviving chemotherapy, they are functionally identical fortresses.
So standard RCHOP chemo fails because it is literally bouncing off a fortress. And it doesn't matter if that fortress was designed by an ABC architect or a GCB architect.
Precisely. The AI saw the fortress. The older genomic methods only saw the architect.
Okay, let's do our second quick recap to pull these threads together before we zoom in even further.
Sounds good.
Bullet 1. By looking at convergent biology, the shared survival tactics, the AI discovered 7 new tumor categories, PG1 through PG7.
Correct.
Bullet 2. PG4 is this highly aggressive fortress that accounts for a massive chunk of early treatment failures, completely ignoring those old genetic boundaries.
Yes, exactly.
So listener, if you were an oncologist today, how would knowing your patient has a convergent tumor like PG4 change your strategy from day one? You wouldn't want to use standard tactic.
No, absolutely not. You'd want to know exactly how that specific fortress operates on the inside.
Which brings us perfectly to the mechanics. Let's take out our microscopes and go inside the PG4 cell. What exactly is happening in there that makes it so aggressive?
Researchers found that PG4 is defined by extreme cell-intrinsic hyperactivity in a few specific pathways.
What kind of pathways?
Well, first, a gene called MYC. MYC is highly expressed. Second, there is a massive overproduction of ribosomes.
Okay, MYC and ribosomes.
And third, there are hyperactive transcription factors known as TCF3 and TCF4, along with intense B-cell receptor, or BCR, signaling.
That is a lot of acronyms. Let's build another visual here. If PG4 is a fortress from the outside, on the inside, it's a hyperactive, totally rogue factory.
I think that works perfectly. Let's run with that factory analogy.
Okay, so in this factory, MYC is the maniacal boss. MYC is standing on the catwalk, face red, constantly screaming, "Produce more! Grow faster! Never stop dividing!"
I love that. And to produce more, you need physical machinery, right?
Yep.
That's where the ribosomes come in.
What do ribosomes actually do?
Ribosomes are the cell's actual protein factories. In PG4, they are the assembly lines working at light speed, churning out the structural proteins the cancer needs to physically expand and grow.
Right. And then you have TCF3 and TCF4 in our rogue factory. These are the corrupted floor managers.
Exactly.
They are down on the floor, taking the boss's orders and executing them, making sure the assembly lines stay open, making sure the B-cell receptors constantly pulling in raw materials.
But, the researchers didn't just want to observe this factory. They wanted to prove what actually sparked it, what caused it to go rogue.
And what did they find?
They found that PG4 tumors are highly enriched for mutations in a specific gene called BTG1.
BTG1. But wait, just seeing a mutation on a spreadsheet doesn't prove it causes the factory to go rogue, right? Correlation isn't causation. How do you prove it in biology?
Well, you have to build it yourself. They used a mouse model.
Okay, a lab mouse.
Yeah. They took mice that already had a baseline cancer predisposition of AVP-BCL2 transgene, to be specific, and they genetically engineered them to also carry the exact human BTG1 mutation they found in PG4.
The exact same mutation.
Yes, it's called the BTG1Q36 mutation.
So they basically installed the corrupted software into a living mouse to see if it would build the rogue factory.
Exactly, and the mice developed the exact PG4 phenotype. The cancers grew wildly, and when they analyzed the RNA of the mouse tumors, it showed the exact same MYC and ribosome hyperactivity we saw in the human patients.
That is unbelievable.
Yeah, the BTG1 mutation essentially acts like someone sneaking in and cutting the brakes on the ribosome assembly line. It just won't stop.
That is wild. But what about the corrupt managers, TCF3 and TCF4? How did they prove those specific proteins were actively on the floor issuing orders rather than just floating around uselessly?
Oh, this is brilliant. They use a technique called single-cell ATAC-seq. ATAC-seq.
Okay, break ATAC-seq down for me. How do you look at a cell and know a microscopic manager is actively working?
It comes down to how DNA is physically stored. Inside a cell nucleus, your DNA is incredibly long. To make it fit, it's tightly spooled up around proteins, almost like thread on a wooden spool.
Right, like a spool of thread.
If a gene is turned off, the thread is wound up super tight. The cell can't read it. But if a gene is active, the DNA has to physically unspool so the cell's machinery can access the code.
Oh, okay.
ATAC-seq uses an engineered enzyme that drops chemical tags exclusively onto the unspooled open sections of DNA.
So you are physically mapping exactly what chapters of the instruction manual are currently laying open on the workbench.
Precisely. And they found that in PG4 cells, the exact spots on the DNA where TCF3 and TCF4 bind were wide open.
Wow.
The managers were actively on the floor, reading the instructions and issuing directives.
That level of precision is just staggering to me. So they have the whole factory mapped, the screaming boss, the assembly lines, the managers, the cut breaks. Did they try to shut it down?
They did. In human cell lines in the lab, they applied experimental drugs called MYC-MAX inhibitors.
Did it work?
Yes. By chemically blocking the maniacal boss MYC from issuing orders, they effectively shut down the entire hyperactive machinery in the PG4-like cells.
Okay, stepping back to process this, time for recap number 3.
Go for it.
Bullet 1. Inside, PG4 cells are driven by a hyperactive MYC boss and TCF34 managers.
Yep.
Bullet 2. BTG1 mutations act like cutting the brakes on the cell's protein assembly line, the ribosomes. And they proved all this using a mouse model and ataxic.
Perfect summary.
So, listener, here's your question for this section. Imagine trying to shut down a real rogue factory. Do you spend your energy targeting the screaming boss, MYC, or do you try to literally unplug the assembly lines, the ribosomes?
It's a really tough dilemma for drug developers, actually.
I bet. But wait, looking at the bigger picture here, there is a glaring logical problem.
What's that?
If this factory is operating at absolute maximum capacity, generating all this heat, making all this noise, producing massive amounts of mutant proteins, why doesn't the body's police force just shut it down?
You mean the immune system?
Yeah.
Yeah.
A factory this loud shouldn't be able to hide. The immune system should wreck it.
And that question brings us to the final and perhaps most crucial piece of the PG4 puzzle. We need to talk about the tumor microenvironment, or the TME.
The TME.
The TME is the complex ecosystem of normal cells, blood vessels, and immune cells surrounding the tumor. Now, in some of the other categories movie I discovered, like PG6, the TME is swarming with active, angry immune cells fighting the cancer.
But PG4 is different, I'm guessing.
Utterly different. PG4 has what researchers call an immune-depleted microenvironment.
Let's expand our analogy. The immune cells, specifically the CD8 plus T cells, are the security guards of the body.
Right.
In a normal immune response, they see the rogue factory, they bust down the doors, and they arrest the mutated cells. But in PG4, what's happening?
It's like the security guards have been forced to work 100-hour shifts without a single break.
Oh, wow.
They are physically present, but they're deeply biochemically exhausted.
So they are just sitting in the guard shack, staring blankly at the monitors, literally waving the cancer cells right through the checkpoint.
That's exactly what's happening. And to prove this wasn't just a theory, the researchers used something called spatial transcriptomics.
Spatial transcriptomics.
Specifically, a cutting-edge platform called CosMx combined with multiplex immunofluorescence.
Okay, those are very heavy terms. Let's demystify them. How do you prove a microscopic cell is exhausted just by looking at a tissue slide?
Well, traditionally, to analyze a tumor, you'd grind up the biopsy. It's like making a fruit smoothie.
A smoothie.
Okay, you know there are strawberries and bananas in there, but you have no idea how they were arranged in the bowl before you blended it.
Right, the structure is destroyed.
But spatial transcriptomics allows you to look at the intact tissue slice. You map the exact physical X and Y coordinates of every single cell while simultaneously reading its RNA.
Oh, so you preserve the neighborhood. You see exactly who is living next door to who.
Exactly.
And where does the fluorescence come in? Multiplex immunofluorescence.
Right, that uses glowing, customized antibodies that lock onto very specific proteins on the outside of the cells. When they looked at the intact PG4 tissue, the fluorescence showed two critical things.
Which were?
First, the guard force is severely understaffed. There are drastically fewer T cells near the tumor to begin with.
And the few guards that are actually there, what did the glowing antibodies show?
They revealed that these surviving T cells were coated in massive levels of two specific surface proteins, PD-1 and TIM-3.
PD-1 and TIM-3, what do those mean?
In immunology, high levels of PD-1 and TIM-3 are the literal molecular definition of T-cell exhaustion.
Wow.
The cancer isn't just hiding from the immune system. It is actively pumping out signals that paralyze the T-cells, putting them into a functional coma.
And this perfectly explains the clinical mystery we started the entire episode with.
It really does.
This is why standard RCHOP chemo fails patient B. Because RCHOP doesn't just poison the cancer, it actually relies on a functioning immune system to come in and clean up the debris after the chemo damages the cells.
Exactly. If the T cells are paralyzed, the cleanup crew never arrives.
Yeah.
The cancer just rebuilds the factory, and the patient relapses.
It is a perfect, devastating storm. The cancer is hyperactive on the inside and completely shielded on the outside.
Yep.
Okay, final recap. Let's bring this section home. Bullet 1. PG4 tumors exist in an immune-depleted microenvironment.
All right.
Bullet 2. The spatial mapping proved that surviving immune T cells express markers, specifically PD-1 and T3, proving they are functionally exhausted.
Spot on.
So, listener, knowing that the tumor actively paralyzes the immune system, how does this change your perspective on therapies that rely entirely on boosting the immune system?
That's a huge hurdle for the future. We have an entire generation of modern therapies, like certain immunotherapies, that rely almost entirely on taking the brakes off the immune system.
Right.
But if the immune cells are already past the point of exhaustion, just taking the brakes off might not be enough to wake them up.
Man, that is intense. So let's synthesize everything we've uncovered today. We started with a mystery. 2 patients, identical diagnoses on paper, vastly different outcomes.
Right.
By moving past just the genomic blueprints and combining them with the proteomic bricks, multi-omics, unmasked PG4, it is a high-risk, immune-evading, hyperactive rogue factory that cuts across all our traditional genetic boundaries.
And the clinical implications of this are just immediate and profound. This proteogenomic framework gives us highly predictive biomarkers.
Meaning we can test for it early.
Exactly. We can now look at a patient's protein profile and identify patient B before we put them through months of grueling, ineffective chemotherapy.
Because we know our CHOP is just going to bounce right off that fortress. So what do we do instead? Where do clinical trials go from here for PG4 patients?
You have to design trials explicitly tailored for the PG4 fortress. That might mean using the MYC inhibitors we saw work in the lab to silence the maniacal boss.
Right.
Or it might mean using multi-targeted combination regimens. There's a protocol called VIPOR, for example, which has actually shown significant promise in tumors with this specific kind of high MYC activity.
So the goal is precision.
The ultimate goal is to use this AI-driven multi-omics approach to route PG4 patients immediately to the exact drugs capable of breaching their specific defenses.
It's a complete paradigm shift. It really makes you realize how incredibly dynamic and adaptable cancer really is.
Oh, the tumors have always adapted.
Yeah.
What's exciting now is that our diagnostics are finally adapting to keep up with them.
It really is exciting. And, it leaves us with a fascinating, almost philosophical shift in how we view medicine.
How so?
Well, listener, think about this. If tumors can convergently evolve, if they can hijack cellular factories and actively exhaust our immune defenses completely, regardless of their initial genetic starting point, are we entering an era where we stop treating cancer based on what it is biologically and start treating it purely based on what it is doing? It's definitely something to think about until our next deep dive.